The KSP Titration Lab is performed by Jasleen, Chrissy, and Kevin. The purpose of this lab is to understand what molar solubility and what KSP is, and how they relate to each other. This can be demonstrated through a titration of calcium iodate with sodium thiosulfate. Okay, first thing you gotta do is go get your goggles on. What you need is 1 molar HCl, starch, 0.05 molar sodium thiosulfate, saturated calcium iodate, water, a pipette, a dropper, a weighing dish, Erlenmeyer flask, and potassium iodide. Don't forget it has to be solid. You also need a ring stand along with a burette and a funnel. Make sure it has a tip. Okay, go and weigh your potassium iodide. You need about 2 grams of it, as close as you can. While someone is weighing the potassium iodide, another one is rinsing their burette with sodium thiosulfate. Basically what you do is you add about 5 milliliters into it, and you rinse it into a clean, clear beaker. And then, once it's been all drained, you add the sodium thiosulfate all the way up until it's 0 milliliters, and make sure it's at 0 milliliters at the meniscus. Okay, after you fill the burette, what you need to do is you need to add about 50 milliliters of H2O to the Erlenmeyer flask. You need to make sure it's the distilled water. Okay, then you add the 2 grams that you'd weighed previously of the potassium iodine, and you swirl it. Make sure it's all in there first. Then you swirl it so that it all dissolves, since it's a solid going into a liquid. Then after you add that, you add the calcium iodate. Basically what you do is you use a pipette and you pull up 10 milliliters. Make sure once again that the meniscus is at the 0 milliliters mark. And put it in the Erlenmeyer flask and swirl. After you do this, you add 10 milliliters of HCl, and that's what causes the dark brown color. Okay, so now we can start our titration. Basically what you do is you add a little bit of sodium thiosulfate at a time, so it's gradual so that you don't accidentally add too much. Okay, once it turns about a light yellowish orange, you add the starch, and this turns it into a dark blue, almost blackish color. What you want to make sure is when it turns clear, that is when the experiment is over. So once again, you're still trading, and look at that clear. So your initial trial was of 19.71, and your second trial was about 18. So now we got to clean up. Make sure you take apart the burette so that you can clean the tip individually so that nothing is dirty and disgusting for the next group who uses it. Repeat these steps for another trial. And thank you for watching our lab.